Without further ado, I am absolutely delighted to introduce you to Professor Oliver Bandmann. Oliver has been leading the charge with a drug called UDCA, and he has been running a clinical trial up in Sheffield, which has also had a parallel arm down here in London. And he's going to share that story with you. And I'm longing to hear the whole story all put together. So without further ado, Oliver, over to you.
Good afternoon, everybody. Thank you, Helen, for this kind introduction, and thank you to all of you for coming in person or attending this meeting remotely. I'm really chuffed to talk about my pet academic subject, a big passion, that's drug discovery for Parkinson's disease, and how Cure Parkinson's and others have helped me to contribute a little bit to this exciting and really necessary field. So let's remind ourselves what this is all about. So Parkinson's disease continues to be the second most common neurodegenerative condition after Alzheimer's disease. We currently only have symptomatic treatment available for a condition which affects at least 145,000 people in the UK. In the UK alone, at least 80 people are newly diagnosed with this condition every day. The currently available symptomatic treatment largely focuses on the dopaminergic deficit. Treatment is only effective for some of the motor symptoms, and the longer a patient is on symptomatic treatment, the more likely they are to develop side effects from this medication. There is no cure. There is no disease-modifying treatment. I do a lot of student teaching, and I often compare it to the situation we have for multiple sclerosis. So when I was a medical student, which admittedly is a little while ago now, but when I was a medical student, we did not have disease-modifying treatment which would slow down disease progression for any major neurological condition, right? But look at what's now happened for multiple sclerosis, where we have at least a dozen incredibly powerful disease-modifying treatments which reduce the number of relapses, which slow down the disease progression. What could we do? When I was a junior doctor, the best thing we could do is put patients on a steroid drip for 5 days. Did it help? Well, it made us feel better, but whether it helped the patients is another matter. So in Parkinson's disease, we're still waiting for these breakthroughs. That's where we are. And apart from anything else, even if you are a politician who happens to be completely uninterested in reducing the suffering resulting from this condition, you should be interested in finding better treatment and funding initiatives to find better treatment, because in the UK, in the US alone, the annual costs arising from Parkinson's disease, annual costs, exceed $50 billion. If we don't manage to get on top of the problem resulting from more and more people living longer, and therefore more and more people having neurodegenerative conditions such as Alzheimer's disease, Parkinson's, and so on, we will break all our healthcare systems and the social systems too. So in Sheffield, we've become particularly interested in mitochondrial dysfunction. What are the mitochondria? They're the powerhouses of the cell. They provide the required energy for all the different functions the cell has to deliver on, so to speak. And it's now been known for a good 30 years that mitochondrial dysfunction is impaired in the brain of patients with what's called sporadic Parkinson's. So these are patients who don't have a positive family history. Oops. There's actually been, the first author on that seminal paper was Tony Shapiro, whom some of you will know, true titan in the field. But mitochondrial dysfunction has also been implicated in all familial forms of Parkinson's, and all identified environmental toxins also inhibit mitochondrial function. So it's very strong evidence that mitochondrial dysfunction, that a malfunction of the cellular powerhouses really matters. But on the other hand, all clinical trials which have tested compounds thought to rescue mitochondrial function have failed in clinical trials for Parkinson's. They did not result in a slowing down of the disease progression. How come? So there are lots of different reasons for this, and it's easy to criticize others with hindsight, And these were different times then, but still. Two key problems, in my view, were that the compounds then were typically tested in inadequate toxin-induced model systems of Parkinson's disease. First major reason. Another major reason for many of these compounds were that they were immediately taken from the bench, so to speak, into large clinical trials. There were no early proof of concept studies to de-risk costly phase clinical trials. In particular, there were no studies which confirmed that these drugs actually got into the brain, believe it or not. And there were no studies which confirmed that there was something called evidence of target engagement. I'll come back to that later. And because of that, there were also no early clinical trials which gave a bit of an idea whether, this drug may actually give you clinical signal or not, a positive signal or not, indicating possible efficacy. So in Sheffield, we decided to use a completely different approach. For the first time ever, an entire compound library with 2,000 compounds was screened directly in Parkinson's disease patient tissue, not in rats, not in mice, not in toxin-induced kidney cells from hamsters or whatever, all this nonsense, but in patient tissue. And we made sure that we would only recruit patients into this, so to speak, or rather ask for their tissue, where we knew that mitochondrial dysfunction was a major factor, because some patients develop Parkinson's because they've got a mutation in a gene called Parkin. And we know, it's been known for a long time, that this particular subtype of Parkinson's is, a group of patients where mitochondrial dysfunction is particularly prominent. That's why we've focused on those. So, and 50% of the drugs we screened were already licensed drugs which were already in clinical use. We're, of course, hoping to identify drugs which we could then readily take into clinical trial. That's called the so-called drug repurposing approach. And further key change to previous drug discovery attempts was we didn't so much look at the effect on cell death itself, because cell death is actually something comparatively non-specific. We tested the effect on mitochondrial function or rather dysfunction, and subsequently then validated the effect in neuronal model system. So this just shows you again how we did it. We first took a skin biopsy. I had one myself. I've got a scar to prove it. Typically from the upper arm. We then grew the skin cells in the Petri dish, and then the cells were stained with a lot of different dyes and so on, and that then really allowed us to do the actual drug screen. It was a multi-stage, really complicated process, and all this work was done single-handedly by a great scientist, Heather Mortiboyce, who was then my postdoc, but has now become a professor in her own right, still works at Citron. That's one of the great things, possibly the single best thing about the Sheffield Institute for Translational Neuroscience. We work in one building. So Heather's office is opposite to mine, and there are other offices from other basic scientists. We just on the same corridor, we bump into each other, we discuss things whilst grabbing a cup of coffee or otherwise, and that just helps so much with the interaction. So this was a long, laborious process, and to cut a long story short, the drug we become most interested in was, of course, ursodeoxycholic acid, or UDCA. So what is UDCA? It's a naturally occurring bile acid. All of us produce a little bit of UDCA, and there are actually some intriguing reports which suggest that there may be something wrong with bile acid homeostasis metabolism in people with Parkinson's in the first place. But it's also been, and some of you may have heard of the gut-brain axis, this intriguing link between the gut and the brain. Nobody quite knows how it works, but there may be something interesting there to do further research on. And it is a drug which has been used for decades for the treatment of liver disease. So liver disease called primary biliary cholangitis, an autoimmune disorder, which again is due to mitochondrial dysfunction. So an intriguing link between Parkinson's disease and this rare liver disease. It's affordable, and others had already shown that it's safe and well tolerated in motor neuron disease patients. And importantly, Clifford Steer's group from the States had already done what's called a PK study, pharmacokinetic study, which demonstrated that UDCA, the more a patient takes, the more UDCA actually gets into the CSF. The higher the levels are in the serum, in the blood, the higher the levels will be in the CSF. So that tells us that this drug in the cerebrospinal fluid, this tells us that the drug can actually cross the blood-brain barrier, right? And therefore stands a better chance, at least, to work in the brain itself. So as I alluded to early on, we've started off by working on patient tissue from the Parkin mutant patient subgroup. We then subsequently went on to more common genetic subgroup, LRRK2 G2019S patient fibroblasts. We also, again, we confirmed the effect there. So LRRK2 related Parkinson's disease is considerably more common than Parkin related Parkinson's. We also demonstrated a beneficial effect in people, not patients, but people who have the mutation already, but have not developed Parkinson's disease brackets yet. Again, they already have mitochondrial dysfunction in their skin cells. And again, we can rescue mitochondrial function there. And finally, we also managed to demonstrate the beneficial effect in mechanistically stratified sporadic Parkinson's patient tissue. But we were by no means the only group working on UDCA and Parkinson's disease. Other groups had sometimes before us already demonstrated a beneficial effect in toxin-induced cell culture models of Parkinson's, and also in transgenic flies and the standard toxin-induced Parkinson's rodent models, the rotanon rat model and the MPTP mouse model. If you know of any compound which has been tested in more or more relevant model systems for Parkinson's than UDCA, let me know. So I thought, oh, this is fantastic, this is such a smart approach. And boy, our results, they match so beautifully work done by others in Portugal and Egypt and the States and in China. And, surely this is going to be, it's just going to be the case tomorrow that the Medical Research Council, the MRC, and the Wellcome Trust and so on, they will bend over backwards. They'll beg me to fund, a clinical trial. Not so. What happened next was the famous valley of death between bench and bedside. That's where so many compounds never actually move forward. They get lost in that desert, and people just can't quite face anymore to put in even more effort to move a drug across that desert. But and I hadn't expected that, and that was a tough time, I have to say. And it then took me 5 years and 7 grant applications until I finally had the money. So I just thought I'd try once more. If I don't get that, then I'll give up. But I did get the money. It's a great shame that John can't be here today, John Moulton. It is his foundation who gave the largest chunk of money, John, from the J.P. Moulton Foundation. But I also really want to acknowledge Cure Parkinson's, not only for the money they provided, but also for the fantastic support they provided for throughout. There are too many people listening from Cure Parkinson's at the moment. I wonder why, but they're really a great bunch. Thank you. Thank you for the support you provided throughout the entire trial. We also had great support from Promed, a company in the Czech Republic, which provided us with the drug, with placebo and packaging for free, and also local support. Okay, so what is this study then? It's the UP study, a phase 2 placebo-controlled. So some of you may not be familiar with these terms. I don't want to patronize anybody, but I just thought it might make sense to explain these terms step by step. Phase 2 means this is a clinical trial which, where a drug has already reached a stage where we no longer need to work out whether it's safe and well tolerated in healthy people. It's a trial which can be and is being carried out in patients, typically looking for early clinical signals. Double-blind means neither the patient nor the treating physician knows whether the patient is on the real stuff, the UDCA, or the sugar tablet, the placebo. Randomized means it's completely random who ends up, and unpredictable, who ends up on the drug, the real drug, the UDCA, or the sugar tablet. And to assess the safety and tolerability of 30 mg per kg daily UDCA in Parkinson's. The design follows closely the design of the exenatide trial. So Dylan is here, Dylan Tauda from Tom Faltini's group. I could not have done the study without the support from Tom and others from UCL. They've been fantastic. And it's treatment for a year with a final 8-week washout period. We recruited 30 research participants only. Yes, it is a small study. Why didn't you include more people? Because we didn't have more money. It's as simple as that, right? What makes you think that 30 is then enough to give you any meaningful results at all? We did lots of clever stats before we actually started the trial. And believe me, we made sure that the trial, was still powerful enough to actually be carried out and then give us a meaningful result in the end. And it was. So we had a 2-to-1 randomization. So 20 patients were on UDCA, 10 on placebo, 2 centers, Sheffield, UCL. And again, I need to emphasize, yes, we knew that UDCA was safe and well tolerated in people who had liver disease, but we didn't know yet whether it would also be safe and well tolerated in patients with Parkinson's. And what's more, people with liver disease only need to take about 10 to 15 milligram per kilogram for the treatment of their liver diseases. We knew that patients had to be on twice that dose to make sure that enough UDCA would cross the blood-brain barrier and would get into the brain. So primary outcome was safety and tolerability of 30 mg per kg UDCA compared to placebo. We also have a bunch of secondary outcomes. I'll come back to that later. Study population, it's most wonderful. I have to say, I keep meeting more and more people who actually participated. I've met with, I think, all of them via Zoom, but I've met yet another, I don't want to reveal who it is, but met yet another wonderful person today who was a trial participant, and I can't thank the trial participants enough. All this happened then, a large part of the study happened throughout COVID, and that was hard. That was really hard. We got the trial off the ground and it worked fantastically well. And then we thought we're flying, and then COVID hit. But we managed to complete it. We did it. And the name of the game is flexibility. So, I managed to change some rules, for our R&D office. But, the name of the game is flexibility. My wonderful research fellow was pulled out of the clinical work to, quite understandably, to do work on the wards. The wonderful research nurse was pulled away from research, quite understandably, to work on intensive care units. So all that made sense, but it's still so bloody hard to keep things going, right? But we did. So I suddenly became the research nurse, didn't quite dress up as one, but, I did what she did. And, it just helped, right? And, so many rules were suddenly changed, and we just kept things going. And we did, and we completed the study. And I'm really proud of that. So the patients we ended up with were quite comparatively young, 56 mean age. The disease duration was really quite short, only 16 months. We defined disease duration as the time since diagnosis. I completely understand that, the disease typically starts earlier, than sometimes there's big lag, but still we had to start somewhere. But why did we concentrate on these patients? So the fact that we ended up with so many young people was, to a certain extent, chance. The fact that we ended up with so many people who only just had developed Parkinson's was not chance, because we, as one of the key inclusion criteria, we wanted to make sure that we would only include patients who've only had Parkinson's for up to 3 years since the diagnosis. Why did we focus on those? Because we thought there is the best chance of us actually rescuing as many dopaminergic neurons in the brain as possible. Results. So key first take-home message is UDCA was extremely safe and well tolerated. This shows you the type and frequency of the UDCA-related side effects. So the most common side effect we observed was diarrhea. It was in all cases mild diarrhea only, typically for only 24 hours, always self-limiting, no treatment required, and not a single patient pulled out of the entire trial because of the side effect of diarrhea or any other side effects of the medication itself. And many of you will know or indeed experience that constipation is, of course, a very common, sometimes extremely troublesome non-motor symptom in Parkinson's. So there's a suggestion that, constipation may be less common in the UDCA treatment group than a placebo group. But of course, the figures are far too small to draw any conclusions here. However, I think it's logical to assume that a drug which has diarrhea as a side effect is likely to have a positive effect on constipation. So, and indeed, I've spoken to so many patients over the last few years about the trial results and so on. And so when they then ask me about the side effects, etc., I say, well, the most common side effect we see is loose stools, and the response I always get invariably is a smile and say, that would be nice. So, maybe we'll manage to help them here as well. So side effects are simply not an issue for this drug. Something else which is really important, some of you may know that some drugs have recently been trialed in proof of concept studies for Parkinson's, where there was very strong preclinical evidence that they should really be promising for neuroprotection. But, when people then did the trial and did the blood tests throughout the trial, there were too many patients with markedly abnormal blood tests, and major safety concerns were raised about these compounds. We did meticulous monitoring of safety bloods throughout the entire trial. Every 3 months, we did multiple blood sampling, and we did not have a single medication-related change in any blood tests throughout the entire trial. So, primary outcome, tick. UDCA is extremely safe and well tolerated, excellent compliance. There's an issue with the pharmaceutical preparation. I'll come back to that later. But it does really have major consequences for the next trial, because this means that, in my view, routine safety bloods will no longer be required at all interim visits of subsequent larger studies. And we will also not need frequent face-to-face visits from the safety and tolerability point of view, which means that trials will, the UDCA trial, and we'll be hearing about ACPD in a moment, the future of trials will be different. For what a nonsense, for example, to expect patients to come back to hospital every 3 months to pick up their trial medication. What a nonsense. COVID happens, and suddenly we manage to ship out the drugs. Well, of course, why weren't we able to do this before? Now we will. So, okay, safe, but what about the clinical effect? And here I cannot stress enough that the study was vastly underpowered to actually really detect a neuroprotective effect of UDCA. This was a proof of concept study. We will need hundreds of patients who will be on treatment for several years before we know whether a drug really does slow down Parkinson's or not. But let me just tell you a little bit about the challenge. So the gold standard, how we assess the effect of UDCA on disease of any drug in Parkinson's disease, is a clinical rating scale called MDS-UPDS Part 3, which stands for, sit tight, Movement Disorder Society Unified Parkinson's Disease Rating Scale Part 3. Bit of a mouthful. What does it actually mean? Well, patients are asked at the beginning of the trial, before they're put on medication, to do this. And then a year later, they're asked to do that. And we're then meant to tell the difference. It's just not very accurate. Of course, it's a little bit better than that. Of course, they need to do a lot more than that. They also need to tap their feet, walk up and down the corridor, do other things. But it is highly subjective. We do not have an objective blood-based biomarker, for example, to monitor disease progression in Parkinson's, which is a huge problem. And if you use this clinical rating scale only, then we did not see a difference between patients and controls, which is something which didn't surprise me in the slightest, I have to say. But the field is now getting very, very interested in a complementary approach, and that's the objective quantification of motor impairment. Many of you will have heard of Fitbits. Some of you may be using them. And this is so the field is so interested in Apple Watches or other devices which would really objectively monitor how well you're moving, how quickly you're moving, how fast you're moving, and so on. And I was very lucky in Sheffield to have a fantastic collaborator, Professor of Bioengineering, Claudia Matzer, who had really dedicated her life to the validation of these biosensors. And all patients needed to do was this. So this is obviously me. Of course, I didn't know that I was being filmed as my cheeky research fellow. He filmed me walking up and down the corridor in our clinical research facility. And as you can see, I was wearing sort of a bunch of different sensors here on the head and the pelvis and here on my ankles. And that really allowed us to get a huge amount of gait-related data. So speed, swing time of the arm for either arm, stance, cadence, stride time, and so on. And this part of the study was only carried out in Sheffield for various reasons. So this is now the most hotly debated result from our study. I'll take you through it slowly. So this is the result of the gait analysis. On the left, you see the placebo-treated group. On the right, you see the UDCA-treated group. And there are always two data points. So the first one is, baseline prior to treatment, week 0, and then after 48 weeks. And every single line depicts an individual trial participant. So please note the very small number of patients included, 6 here on placebo, 12 on UDCA. But what you see and what we looked at is cadence. That's the number of steps per minute. And what you can see here is that the number of steps per minute decreased over 1 year in the placebo-treated group in pretty much all participants bar one, whereas in the UDCA-treated group, the number of steps either stayed the same or went up in about 2/3 of all patients. In some others, it went down. You could then say that patients on UDCA, on average, take an extra 6 steps compared to patients on placebo after a year of treatment. Now, I think this is interesting. Does it prove that this drug is neuroprotective? No, of course it doesn't. Does it hang together with other data we've got? Yes, very much so. So, for example, the patients suddenly walk more symmetrically, and their overall walking pattern becomes more sort of fluid, etc. So all of this, I think, fits to the assumption that UDCA probably does something good in these patients. We will be seeing much more similar data sets in the future. It will be extremely exciting to work out what these data sets actually mean. Okay, so how can we make sense of this discrepancy between the clinical rating scale and the objective quantification? And again, I want to emphasize, when you start a trial, of course, you hope that everything will match up nicely. It's just not the case. This is science. It's real life science. And, you just got to find a way how you can try and make sense of discrepant data sets. And if the objective sensors were only as good as the clinical rating scales, we would actually not need them. So, but I just wanted to highlight again the level of granularity we get from the clinical rating scale and from the objective quantification. So here is just the gait item of this clinical rating scale, and all our patients were either scored as 1, slight impairment, independent walking with minor gait impairment, or 2, mild, independent walking but with substantial gait impairment. Where do you draw the line here? And all the 18 patients included, indeed, all the 30 patients included in the entire trial, were either stage 1 or stage 2, and, only 1 from each group moved to the other group, if you see what I mean. Because it's such a marked change you would need to fulfill, so to speak. You would need to observe to actually justify that different scoring. Compare that to the wealth of data we get from the sensors. So data from at least 50 steps, data collected with sampling frequency of 128 Hertz, with a spatial resolution of 1 centimeter and a temporal resolution of milliseconds. Of course, we will be getting hugely more detailed data. how much they really tell us about disease progression in Parkinson's disease is something we need to confirm. But it is, I think, the right approach to combine these approaches. Another problem we had not predicted at all was that some of the research participants were just absurdly fit, unbelievably fit. So, these are all photos, of course, I got permission from all of them. So this is a wonderful, wonderful gentleman who's still doing completely mad time trials. I love cycling and all the rest of it, but, never in my life was I half as fast on my bike as he still is with his Parkinson's. And this is another gentleman whom some of you might have by now recognized. He's in the audience, who's done completely mad bike rides a couple of years ago. And he was in the UP study. And then there's another lady whom I think I haven't met yet in person. She still does water skiing, And I think I'm getting sidetracked, I often do, but I think it's really important that we get the balance right, because every patient here, every relative of a patient with Parkinson's here in the audience knows more about Parkinson's than I ever will. I really mean this. But I cannot fathom, your daily struggles. But, it's also really important that we don't stigmatize patients with Parkinson's, right? That we really always actually also emphasize, what they can still do, and I've got such a fantastic collection now of photos like this. So, but, okay, focus, Oliver. So, but, I think in some ways, and I hope these wonderful participants will not feel offended when I say this, this was actually a problem for us, because I personally think that these rating scales are accurate enough, especially in patients who are very much at the high functioning end of Parkinson's disease, right? I just don't think that the UPDS Part 3 rating scale is accurate enough to take small changes, maybe especially in highly functioning individuals like these three and so many others. It's a huge, huge challenge for the field. Okay. Let me move on to the final data set I want to share with you. The single biggest mistake I made when I tried to develop a clinical trial protocol for Parkinson's, when one after the next grant application got shot down, was I didn't quite understand what target engagement actually means, and I didn't quite understand how important it is. It took years until the penny finally dropped. So what this means is, it does not mean that you find evidence that drug has actually crossed the blood-brain barrier and swims around in the cerebrospinal fluid. What it really means is that you need to find a way, one way or another. It doesn't matter which tool you use. Different tools will be the right thing for different disorders, for different drugs. That you will need to find a way to show that the drug you're testing in Parkinson's really does in the brain of patients with Parkinson's the same thing as it did in the Petri dish. In the context of UDCA, we were very interested to get any evidence that UDCA really rescues mitochondrial function in the brain of Parkinson's patients. And we wanted to make sure that we would get this proof from the area where it matters most. And that's the brainstem, the midbrain, where the substantia nigra sits, where, of course, most of the Parkinson's disease cells die. So, and the way we did this, and again, I was lucky to have excellent collaborators in Sheffield in the imaging, the radiology department. It was a special type of MRI called phosphospectroscopy. So the patient will experience a 31 phosphospectroscopy scan, just like a normal MRI, but we get extra images from it, which essentially allow us to depict, to measure energy-rich metabolites in the brain. Okay, I've tried really hard to avoid technical lingo right throughout my talk, but I will fail at the last hurdle. So I apologize. I didn't quite know how I could have used different terminology. Let me try. So the first thing I want to say is take-home message is, yes, we did actually get evidence that UDCA treatment does rescue mitochondrial function in the brainstem. How did we do it? we found significant change of something called Gibbs free energy of ATP hydrolysis. So the way the cell can make good use of the energy provided by the mitochondria is by hydrolyzing ATP. In some ways, it's like burning fuel, right? So, and delta G ATP expresses the amount of energy released from hydrolysis of ATP to ADP and inorganic phosphate. More negative values reflect an increase in ATP hydrolysis. So on this figure here, what you see here, that's placebo and that's UDCA. And every point here depicts a patient who was on placebo, and it depicts the difference in the levels of Gibbs free energy between baseline and a year later. And remember, positive values are actually bad, right? So less energy is released, and negative values are good. So more energy is released in UDCA. So there's a clear statistical difference after a year. The figure for the Gibbs free energy in all the patients has gone down, right? So more energy is being released, but in the placebo-treated group, the values have gone up. So, and that is in the midbrain. That's for the first time ever. Nobody's ever managed to do this before. So, summary. UDCA has got an excellent safety and tolerability. There's a reliable PK profile throughout. I've not shown you these data. There's consistent improvement of gait parameters across several different domains, and phosphospectroscopy provides at least tentative evidence of target engagement in the midbrain. So what are the next steps? I very briefly told you early on that, you know, there's an issue with the preparation with the tablets of UDCA. We had 2 patients drop out, not because of side effects of the UDCA, but because they simply couldn't manage to swallow the medication. And that in some ways is not surprising. So these are not one-to-one real life image sizes, of course, but you know, these are the trial tablets the patients had to take. You know, these torpedoes, most patients had to take 10 of them a day, in addition to the medication they had to take for the Parkinson's or other illnesses anyway. So, you know, that's a lot. And I was actually amazed that, you know, we had, in the UDCA treatment group, we had a compliance of 97%, right? So it's absolutely phenomenal. I can't thank the patients enough. But drug development is an iterative process, you know, and you always try and look for things, well, what could you improve, you know, with any drug. So I've subsequently managed to secure supply of a different formulation of UDCA. So it's exactly the same substance, but compressed into, you know, a different tablet. And as you can see here, so the new drug, so to speak, we want to use for the next trial is considerably smaller than these torpedoes, but has twice the strength and is also scored. So it should be much easier for patients to take this. These are just some other preparations. I think we should give that another miss. But I just thought it might also be useful to compare that to standard Parkinson's drugs, which may be very familiar to some of you. So that again is the drug we want to, the tablet we want to use for future trials next. Also for like 500 milligram, that's an entacapone tablet, and that's a Modipar CR tablet. And, you know, if anything, Osify is a wee bit smaller. I appreciate these tablets still have a size, but it seems as if, you know, I think this is just no longer half as much an issue as it would have been for the last one. So what are the next steps? So I'm particularly excited. I think it's really wonderful that we'll be hearing another talk by Georgia in a moment about ACT-PD. So I don't want to steal her thunder. For me, ACT-PD is the single most exciting development in the world of Parkinson's disease trials since the very first meaningful disease modification trial on Parkinson's disease more than 30 years ago. I really mean this. But I'm really excited to share the news with you that, you know, UDCA has been chosen as one of the top 3 drugs to be trialed in ACT-PD, brackets, if we get the money, but we will. 400 patients will be put on UDCA, 100 on placebo. The treatment duration for 3 years. The primary outcome will be patient-reported, right? We want to hear from you whether you feel that UDCA has really made a difference or any other drug tested. And there will be multiple secondary and exploratory outcomes. So that's one way how I want to take UDCA forward. But there's also another way, which is also very, very interesting, exciting. So you may know that Parkinson's disease doesn't develop overnight. There's a prodromal period. And we increasingly think that it doesn't last just years, or it may last for more than that, possibly decades. And in some people who will carry on to develop Parkinson's, there are non-motor abnormalities which will make it easier to identify them. So, for example, you may have heard of a Cure sleep disorder called REM sleep behavioral disorder, or RBD, or some people can develop constipation years and years before they develop proper Parkinson's, or problems with a sense of smell. So we call that the prodromal, the pre-motor phase of Parkinson's. And very recently, a blood test has become available, which appears to actually help us a great deal to identify these patients reliably. And there is actually strong evidence of mitochondrial dysfunction already in this pre-motor phase of Parkinson's, which again suggests that mitochondrial dysfunction is a key driver of Parkinson's disease. And I would just absolutely love to also take UDCA into prodromal pre-motor Parkinson's disease. There are lots of people I'd like to thank. I've only listed some of them here, but I'd particularly like to thank Tom Payne, fabulous clinical fellow, who's now an NIHR-funded clinical lecturer, Tom Faltini in London, and the rest of the study team and the funders. Thank you so much.
Oliver, thank you very much. My goodness, that was a whistle-stop tour through a very complicated series of steps that you had to go through to get UDCA into trial, and then actually through the trial, even in COVID years. So incredible, absolutely incredible. I'm sure we've got questions from the floor, but also online. So any questions from the floor for Oliver? Jodie.
Hi there. Did you see any correlation between the patients who performed better in the Gibbs free energy test and those who did better in the gait test?
Yeah, really good question. So, of course, we're very keen to look at that, but we got told off by our statistician because there's something here, a pitfall you need to avoid, which is called overfitting of data. And of course, we still had to play around it, but we didn't really see a correlation. But I'm not too, so that would have been absolutely fantastic, but we didn't see that. Yeah, but we probably, as a statistician told us repeatedly, shouldn't have even tried.
I think Suzanne's got a question online. Sorry, she's behind the scene with a laptop.
Hi, can you hear me? Sorry. Yes, could you please, Oliver, just explain what the ethnic mix was, please, on the UP study?
Oh, and I'd like to know about gender balance as well. That would be good.
So, ethnic mix, really good question. So, short answer is not very good, right? The vast majority, I can't quite remember if it was every single patient, but I believe it was every single patient, was what one could probably easily describe as white Caucasian. And it's a huge problem the whole field is facing. We're trying to do a lot of work on this in Sheffield, but I appreciate there's also work going on elsewhere. We could talk about this for a long time. I think this actually is a problem which goes way beyond clinical trials in Parkinson's. When I look around the streets of Sheffield, it isn't just white Caucasians I see, right? There are lots of people from, with background from what I would describe as Indian subcontinent and so on. But I don't see them in my clinic. There must, and I don't get this. And we're currently trying to do a string of community-based events where we raise awareness for Parkinson's. But yeah, so the short answer is appalling. Gender. I should have read up on this. I can't remember. You will all know that. But I have.
Don't worry if you can't answer it. Actually, I wondered if Nicole could come in on the mix and what we're trying to do as a field to collectively involve a broad range of people with Parkinson's. Nicole's head of patient involvement at Parkinson's UK.
So sorry, just here's the answer. So we had 70% in males and 30% females in the UDCA treatment group, and slightly different split in the placebo group. So you will know that Parkinson's is somewhat more common in males than in females, but in men and women, but it's only sort of something like 3-2 split. Sorry.
Nicole.
Oliver. I actually had another question, but I'll quickly answer this one. So Parkinson's UK, alongside other organizations, are working on race equality in research. So we have a steering group, and we've held a series of community events, which is a great opportunity for studies like this to be promoted at. We've got a series of events happening next year, so that could be a perfect opportunity to raise awareness of studies like this for recruitment purposes. So that's the way in which we're approaching it. The other question I had was around the issue with UPDRS used in sort of early Parkinson's and the sensitivity issue, the granularity. We're working with UCB and Parkinson's Foundation, and we've developed 2 new PRO instruments. And I was just wondering, for the new trial, will you be exploring adjuncts to UPDRS and objective measures because of the issue with obviously seeing whether there is, for disease modifying therapies, whether there is actually a difference when UPDRS doesn't have that level of sensitivity, which has been noted by patients and researchers and so forth? Thank you.
So let me just comment again on the EDI issue. I think it's really important in some ways. I think the single most important thing is really that we all work together in this space. One could argue that maybe all of us ignored it a little too much in the past, but we've been helping the local branch of Parkinson's UK, WANO, and others with 2 community-based events. I've spoken with both. We had the last one at Sheffield University, but for next year, so Emma is here. She actually organized the meeting for them. But next year, we will actually be holding events in the communities, right? Because we need to get out of the universities. We need to go into the mosques or whatever, right? Into community centers, right? And talk to people there. So that's the next big step. And with the census, so we will most definitely use the census for any future trial. The big problem is at the moment, though, that everybody and their dog have their own sensor, right? And we're all quite obsessed with, oh, mine is the best. No, mine is best. Oh, I've got an iPhone up. Oh, yeah, that's great. But, you know, and what charities should do with respect is actually fund really good research which would compare the different sensors to each other. And that's not happening enough. And I personally think that would be the role of Cure Parkinson's or Parkinson's UK. We're in the process of setting up a translational research coordination across the UK. One work stream will be, for Parkinson's, of course, one work stream will be led by Lynn Rochester, who knows more than anybody else I've ever met about the use of sensors to monitor disease progression in Parkinson's, and we'll hopefully be able to do some of that work together with Lynn. It's absolutely crucial.
Can I just come in on the point about the PROs and being able to test those? I certainly know that Camille feels very passionately that the EJS platform can be a vehicle to test these new instruments as they come through. I also just wanted to come back on the wearables piece. There's a fantastic bit of work and a whole work stream that is being coordinated with the Critical Path for Parkinson's and Michael J. Fox Foundation, involving Parkinson's UK and us and Parkinson Canada and Shake It Up Australia, to really look at endpoints. Yes. And to make sure that we are all agreeing what we yes. measure, how we measure them, yes. and also as part of that, evaluating these different approaches. Because we, you know, for me, certainly from my experience of working with the Sense Park project back 5, 6, 7 years ago now, high quality algorithms accurately measuring aspects of Parkinson's should be device agnostic. It shouldn't be about the devices. It should be about what you're measuring and how you're measuring it, and the quality of the data that you can collect. And then that consistent data analysis. So I think there's a huge amount that is ongoing around this.
No, completely
even if
agree.
It's not apparent.
it's hugely exciting, and it needs an international collaboration. And of course, I know a little bit about that collaboration, but it's great, to have that emphasized again. What I really want to emphasize, though, this is not a trivial task, and I know you know this, and I know Helen knows that very well, and everybody works, because again, when I started this, I was completely naive about it, and I thought we'll get a simple readout. It's absolutely not simple, not trivial. It really matters, as Helen says, you need to be device agnostic, and you also need to, I think it's fundamentally important that, whoever works on this has complete access to all the raw data. The big problem with any collaboration with commercial partners is that you only get the readout, the final analysis. And, and some of the devices which are being propagated have actually been developed for something completely different. So it's desperately needed progress, and it is the future. I have no doubt about that, but it's complex.
Oliver, Simon, let's take one more question from the floor. If that's, maybe we can just squeeze in two. Simon, sorry, you're not concerned. Paul, I'm sorry, Paul. Do you know, I nearly always see you together. That's some of the problem. Anyway.
You mentioned the valley of death, and a lot of projects were failing to get across that. Is the research community sharing learning on that?
Well, I've certainly been whinging a lot, but no, just to be serious again, I think things have changed. I think people are now more prepared to fund early clinical trials than they have been in the past. But I think good things are happening, but there need to be more joined up thinking. So ACT-PD, as I said, it's an absolutely fantastic development, but that's for phase 3 trials, phase 3 drugs, right? Drugs which have already shown promise in smaller trials. The next big development we need to have, and I spoke to Simon about this area, is a platform trial design to de-risk phase 2 ready drugs, right? Where we can quickly assess drugs such as UDCA, and lots of them are coming through now. And really, it's just, put the patients on, on treatment for 3 months, and then see whether you get a biological signal, right? Do you get any evidence that, UDCA, I'm just using that as an example, gives you a biological signal which suggests that it really does something in the only model system which really matters, and that's a human patient.
I think this is the final question we're going to take just before the break. Actually, this.
Just want to ask on your patient selection. So you basically say you only use the mitochondria basically patients. So how do you identify who they are and what?
So I'm sorry if I didn't explain this correctly. So we did not, at the moment, we don't have any tools. So what you're really referring to is, are we at a stage where we can apply personalized medicine approaches to Parkinson's disease? And again, that's something we and many others have been working on. The buzzword here is mechanistic disease stratification. Find out whether Peggy Smith, who's developed Parkinson's last year, has developed her disease due to mitochondrial dysfunction or lysosomal dysfunction or alpha-synuclein aggregation or inflammation predominantly, right? All these things will overlap. At the moment, we haven't got the tools to do this. The only way we selected patients was based on how long they've had the disease for. The clinical trial. The preclinical work was done on patient tissue where we just knew that there was mitochondrial dysfunction.